Okay. Welcome to Capstone Podcast.
Glad to be here.
Yeah. So today I have Dr. Waddell, who is one of the founders of RIP Capstone. So yeah, just start about the origin story.
Sure. Actually, I was thinking back to when I was a student here, and we had something called senior thesis. This was back in 1998, 99, and I had a partner, and we went out and mapped all the trails. This was before GIS, and wrote up something about the history of the park and the geology and all the plants that we identified out there. And I think when I came back here, in part, it was for the park. I think that was really formative for me, and I wanted to give that to the students as well. I mean, this is such a special place. And so when I came back here in 2011 and started teaching geology courses, I was contacted by Steve Rudd, who was the natural resource manager at Hot Springs National Park. And he was also a geologist, and so he took me out all around the park and showed me where they'd identified the recharge zone for the hot springs and all sorts of things like that. And he started talking about how he really wanted the park to be more involved with our students and vice versa. There had been some projects kind of, like mine, and there had been some archaeology projects, Mr. Thompson, and some things Dr. Ruley had mentored, but it had been sort of scattershot. And so this was an opportunity to kind of have a formal agreement with the park that we were going to have this program and share resources.
Yeah.
And so that's how it got started. And then once we signed the agreement, it was just a matter of figuring out where to put that into the ISMSA program. And so I teamed up with Dr. Ruley, who previously taught biological sciences. And I'm being a geoscientist, we felt like we made a pretty good team there in terms of what we could mentor in the park.
Definitely, yeah.
We started a Wednesday afternoon wasn't really a capstone course yet, but a Wednesday afternoon seminar course. And we took on juniors. Mostly only seniors were doing science fair projects at the time. And just in those Wednesday afternoons, we taught them all about the park. They were doing their work service for the park, so they were involved on all different sorts of different levels with the park. And yeah, so they managed to come up with projects by February.
That's pretty quick.
It was.
Especially for like juniors.
Yeah, and so in some cases their project work counted for work service, so they were sort of getting that double time in there, and then they were giving back to the park at the same time. And so they ended up placing really well in a lot of categories against the seniors, and I think administration started to take notice of that.
They were like, what's going on? These are juniors. They're not even seniors.
Yeah, exactly. So from there, we started saying, hey, we'd like to move this into the course schedule. We'd like to actually meet, the amount that a class would meet per week. And so the next year we did that. And the students, we felt, did even better. And at that point, the administration started saying, hey, these students are placing and doing well at State Science Fair. Maybe we could open this up to have more juniors work on projects rather than keeping it a senior thing. And they could have things for their college applications and so on and so forth. And we liked the idea of working with juniors because it gave us 2 years. They would continue to do work service for the park, really, junior and senior year at the time, and that gave the park kind of that continuity. Mm-hmm. And so, the park, they let us feature our science fair posters and give presentations at the Fordyce, the student project, so it was just such a great opportunity for our students, and all their papers went in the park archives. And
oh, wow. Yeah. So I didn't know that, actually.
Yeah, so you can contact the archivist and access any of them. Yeah, we've kept that going. So that was when we started. It was 2014, and now we're obviously 2025. Dr. Ruley, actually, in 2016, so only 2 years after we started the program, the park natural resource manager at the time, Shelley Todd, she nominated our group for the Hartzog Award, which is the big volunteer award that the Park Service gives. And so we won the region, which is several states, and then
oh my gosh,
then we went
several
on
states.
to win the national award for the volunteer service by a youth group. So we actually took a group out to Washington, D.C., about a dozen students who'd volunteered and done research in the park. And we got to tour the Capitol building.
Oh my gosh.
And be presented with the award by the director of the National Park Service at the time. So that was only 2 years in. So that sort of validated that what we're doing is not just important for our students, but for the Park Service as well. So yeah, that kept us going. The complicated thing about it has always been the field work, the logistics of taking students out there.
Permits.
Yeah, permits for the park. But we've managed to keep that going. Dr. Ruley passed away in 2020, but now we've got Dr. Dodd, who's an excellent biologist as well, and a field biologist. The program's still going strong. We feel like stronger than ever.
Welcome to Capstone Podcast. And here is our guest star, Kira Burnett.
Hello, I'm in the Research in the Park capstone here at ASMSA.
Or RIP. That's what everyone says.
That's what everyone calls it, RIP.
Yeah. So let's start from the beginning. So how did you come up with your RIP project?
So my project this year is a continuation of when I came in as a junior, because all Research in the Park students have to present their junior year and their senior year as well. So in my junior year, I selected Hot Springs Creek as my muse, you could say, because it's in the heart of Hot Springs and students have had done previous research onto it. So I'm not just going in blind without any research or any knowledge of problems with the stream. So what I wanted to do was to compare Hot Springs Creek, its more urban sites, and then it's less urban sites, more residential or forested sites. And I found no significant differences between the urban and the quote-unquote less urban. However, throughout the entire stream, I found high phosphorus. And high phosphorus is a nutrient in streams. While it's needed for plant growth, it's needed for animal growth, it can be very detrimental in high concentrations, which is what we were seeing in the stream. 0.1 milligrams per liter is the limit for phosphorus in a stream to prevent eutrophication, which is when there's so much phosphorus that it grows huge algal blooms, if you've heard of those, on the...
No, I've never heard of that.
You've
what the
never
heck?
Okay.
You think I watch Animal Planet or something?
It's not on Animal Planet. The Gulf of Mexico every year during the summer has a dead zone caused by eutrophication where it's millions of fish die.
Oh.
And then also off the Gulf of California, or off the coast of California, there's been huge issues, I think back in 2017, 2018, where there's been harmful algal blooms, not just because they deprive the water body of oxygen, but they can contain neurotoxins and they'll kill seals. Seals will have seizures and stuff, or lose their sense of direction and die.
Wait, what's this called?
These are called harmful algal blooms, and they can cause either neurotoxicity or eutrophication, which is a lack of oxygen. That's a little off topic, but you see, I came in with knowledge of that, and I came in with wanting to do something about algae and phosphorus, and so I looked at Hot Springs Creek, saw that it had high phosphorus last year. Now I'm to this year's project, which is where I used a mathematical model called nutrient spiraling to see how far the phosphorus was traveling. I found that model by talking to my mentor, Dr. Alan Dodd. She's a stream ecologist as well. Yes, go Dr. Dodd. So she has a lot of experience with that, and so she could point me in the right direction. And then I also looked at the macroinvertebrates of Hot Springs Creek, since they're important bioindicators of stream water quality. And then an entire other tributary or water body was Bull Bayou that I also looked at for phthalates, which are a set of plastic pollutants. Bull Bayou was sort of out of nowhere.
I'm looking at the map. Wait, no, it's not on the map.
Yeah, see, I have my main map, right? You have David Walking Parks. Bull Bayou is entirely on the other side of Hot Springs, and I have it because it's another tributary to the Washita River, and that's the main concern, because Hot Springs Creek on its own, while it's bad to have poor water quality in a stream, what it drains into and how it's affecting that is the main issue, because that's how you get issues like the Gulf of Mexico dead zone. So I looked at Bull Bayou because if these set of plastic polyurethane phthalates were in there, then they could be making their way into the Washita River. So that's how I got that idea. Dr. Waddell recommended me about that.
So the creek is feeding the Washita River and the Bull Bayou is feeding the Washita River.
Correct.
Okay. And then, so like, you're doing samples. So like, how are you testing for phosphorus?
Okay. For phosphorus, I just take water samples in these conicals, plastic conicals. I will go out, usually I drive to all my sites, and then I hike about a mile or two.
That's a lot.
Yeah, it's bad for some of the sites. Like my furthest downstream sites, I'm like basically in the forest. I have to jump across this one like small stream. It's dangerous. It's treacherous. But I go out, I take samples in these conicals, and then I come back to usually the lab here on campus. I've done it at other laboratories as well, but on campus, we have the machine needed, which is a spectrophotometer. And so I will take a powder pillow, which is like a little packet, sort of like a salt packet, and you put that in your sample, and it will help you test for phosphorus because a spectrophotometer works by like shooting light into your sample, and then whatever reflects back off those particles is how much of like X material is there. It will give you absorbance, and then you can turn that into milligrams per liter of phosphorus.
Because like we were on the site at that one place, right? Mm-hmm. And so like you mix like salt and water, and you called it like a slug.
Oh yeah, that's for the spiraling. That's for spiraling.
Okay. So totally different. My bad.
Oh no, you're good. I mean, that's why I'm trying to explain it. It's like there's a lot going on, but in nutrient spiraling or in phosphorus spiraling, you have to test for the phosphorus as well. Like that's how you're doing for phosphorus spiraling. Grace went out with me, and what we did was we mixed up a bulk instantaneous addition, which is also called a slug. I really haven't figured out why they're called slugs. I guess it's like, you know, like a baseball. you're like a slugger. You're like I never thought about it.
I just thought it looked like the Reds do like slug sleeve. It looks weird. It's
it
like
might be
white, foggy, like, and then it's like in this bucket, and you just like dump it, and then you run and try to chase it. Low-key.
So we mixed together salt, which will peak the conductivity of the stream, the specific conductivity, which is how many ions are in the stream, and so that's how you like know where the slug is coming, because it'll go up to like 120 microsiemens per centimeter. I hope that's the right units. And then it'll go up to like thousands, and you're like, well, okay, now you know the slug is here. But you use salt and then dipotassium phosphate, which is it's a form of phosphate that can be safely administered into the stream. You try not to do this often because, I mean, you are adding a ton of phosphorus into the stream. It has to flush out relatively fast, so you only do these once or twice. You don't do them a ton of times and then like find the average, which is what you try to usually do with other types of sampling.
Yeah.
But for this time, you only do it once. And so we release this slug, and then we take measurements at multiple transects downstream from our release site. And that is to calculate uptake length, which is, it's so like, as you go further downstream from this transect site, you know how much it's decaying or being taken up, and from that you can calculate the exponential decay rate, which is the uptake rate. So you're just seeing how much it's falling out, how much is being taken up over like this set amount of time that we were observing. That's why like Grace and I, we were running, it's like, okay, 10 meters, 15 meters, 20 meters, 100 meters.
All the way to 200.
We went all the way to 200.
It was really cool. Like, I felt like I was, like, chasing a ghost. I was like, where's the ghost? Like, you got the machine. You're, like, looking at the numbers. I was like, what do I do? What do I do? Kira, am I going to mess up? Like, it was really good. And then, so, like, you know, you take most of your samples, like, in-house at ASMSA. And the reason, before we started recording, you said that the reason you went to other labs was because the number was so high of phosphorus?
Yes. So previously last year at my furthest upstream site called David Watkins Park, during storm flow, which is like right after it's rained, or really you're supposed to go out as it is raining at the peak of the storm, or the rising limb, which is like before the rain has stopped or gotten to its like worst point.
Yeah.
We went out and sampled it, and when you add that powder pillow into the sample, it will turn blue. And that's because it has ascorbic acid in it. It will create a reaction that makes ascorbic acid. And so it's a colorimetric process as well. Which basically just means...
No idea.
It just means that it will change color depending on how much is in there. And usually, it's like, a little blue, maybe a lot of blue. But this sample, it was like navy. It was opaque. I have a photo of it, and Dr. Dodd was there with me because we were trying to get these tested very fast. And I don't hear her cuss a lot, but she cussed. She's like, I won't repeat it, but she was like, oh my gosh. And we were like freaking out. And so we're like, we tested it and we're like, this is crazy. It was 3.76 milligrams per liter, which, remember, the limit is 0.1.
Oh, yeah, because it reminds me of, like, Chernobyl, and, like, they're testing the radiation, and it's so high, like, it's past the meter's, like, level. And
That's what happened at first.
They're like, oh, that can't be. Like, there's no way.
That's what happened at first. For my first sample from that site, it just said larger than 2.5 cannot be calculated. And what we had to do was then switch to absorbance and then do it like a calibration curve. So like then you make like I know this has 0.05 milligrams per liter.
That is so high.
I would like sample it out. So if at first it did break the machine, it literally could not tell us how much was in there. And so it was like, okay, let me make a calibration curve, and then it was even higher. And so I thought it was broken, and so we went to another place. We went to the Davidson wastewater treatment plant. Shout out, really good place. If you get to tour it in environmental science, I really liked it. I went there, and they helped me use their spectrophotometer, and everything was right. Like, we tested the same samples at each spectrophotometer, the one at ASMSA and then the one there, and they were the exact same. And so we're like, okay, this can't be broken.
This is too much. Too much. Bad urbanization.
Yes.
Ooh. Okay, and so then like there's multiple parts to your project.
Yes.
You also have the microinvertebrate sampling. Could you talk?
Macroinvertebrate sampling.
Okay, my bad. Could you talk more about that?
Yes. So macroinvertebrates are little buggies like stoneflies, mayflies, like little nymphs, and stuff that looks like worms. Worms as well. Worms are also considered macroinvertebrates. They're the primary consumers of the stream, so they're going to eat each other. They can also eat each other. They can be secondary consumers. Yeah. But they also eat algae, and they're important bioindicators of a stream. So they're like organisms that can tell you what's going on. They like rapidly respond to changing factors in the stream. They also have short lifespans, so they can rapidly like die and live, and can tell you in like just a couple of months if something's going on in the stream. Because there's pollution tolerant species and pollution sensitive species. And so I looked at their levels of pollution tolerance using two different indexes or indices to test for those levels of pollution tolerance. I compared them back to 2020, which was the last time a student did a survey.
And 2020 it was like really diverse.
Yes.
And 2024 it's like just the pollution tolerant, or it's like a lot.
It was majority pollution tolerant species. Like back in 2020, I used the Hilsenhoff biotic index and then the Ephemeroptera, Trisoptera, and Plesoptera ratio, which is just like Hilsenhoff biotic index is the more pollution tolerant species you have, the higher the score. So higher scores are bad.
Okay.
And then for the EPT, those are 3 orders of pollution sensitive macroinvertebrates. So you want more of those because those are better. So those are your stoneflies and mayflies. And in 2020, it was mostly the mayflies, stoneflies. It was really good. You had a lot of pollution sensitive species. But then in 2024, I only found one. I only found one guy out of like 60 that I sampled. And the last sample had like, theirs was 85.7% and mine was 0.01%.
0.01%.
The graph looks so goofy because it's like 85.7 and then it's literally like you can't see it, so I have to zoom in, and I'm like,
that's
there
crazy.
It is.
like you can't see it from afar, but trust it's there. The 0.
Yeah, it's minuscule. It's tiny.
Yeah, that is like kind of insane. And then you find this results and you're like, oh my gosh, like our water, it's thumbs down.
So bad. It's so bad.
It's gonna kill like these ecosystems. And so like, what did you do?
So I then had to I have to write my discussion and conclusion, of course, but it was a lot of talking with Dr. Dodd because I mean these results are dastardly. These are they're pretty bad. Last year I reached out to the City of Hot Springs. I reached out to the Stormwater Division, which is supposed to handle wastewater treatment, as well as the water bodies here, storm drains, anything to do with the city and water. And I reached out to them, and they did not respond. I emailed like 3 people. I called 3 people. I even know where their building is, because their building is the transportation depot, which is known as Broadway Terrace as my sites. It's the same place.
Yeah.
And so I went to their building, and I could not find them anywhere. I was like, I couldn't find their office. I don't know if they exist. Like, the email, it didn't bounce back. Like, I know the email exists. Mm-hmm. And the phone call was like, leave a voicemail, and I did. But I never got anything back. And so this year when I found my discussion, or like when I found my results, I haven't reached out to them yet, but I'm hoping to. And if I'm going to reach out to the city again, because I feel bad going straight like to state, especially if I didn't tell them. Mm-hmm. But there's also the Arkansas Division of Environmental Quality, and they handle, like at a larger scale, they handle impaired water bodies, and they have like a list of impaired water bodies called the 303D list. And I want to reach out to them and be like, hey, can you look at my results and maybe have some of your people come out and check Hot Springs Creek?
Verify.
Or verify my results, and or maybe like talk to somebody at Hot Springs, because I've tried and they won't respond.
You're like, hey, you're like the boss of them. Like,
you're
they'll
like
listen
the boss.
to you. Like,
you can maybe tell them to. I don't even know if these people. I don't know. I'm so up. How can you not respond to an email in a year?
A year.
It's been a year. And one of the places, there's like a site that I'm concerned that could be a source of pollution of phosphorus into the stream. And
the landfill.
No, that's
not the landfill.
Hold on, this one's like
okay. Sorry.
Oh no, you're good. It's like a very small place in my discussion on my poster. You wouldn't notice it. I usually tell it at like when people ask. It's called the Hot Springs Lift Station, which all the
Hot Springs what station?
Lift Station. Okay. All the sewage lines in Hot Springs are gravity based. And so what a lift station does is, as you've got this sewage moving down, it will just lift it back up again. And so it can use gravity to go eventually to the wastewater treatment plant. But when they're all underground, but when they get old and when they get overwhelmed with like a lot of stormwater, sometimes they can leak and, get to the soil and then maybe get into the stream. I was like, well, let's just reach out. Let me see if I can find any other records. When's the last time they had anyone look at this?
Yeah.
I have no evidence besides Google Map and literally being there that I know exists. It's not anywhere on the City of Hot Springs site.
That's kind of crazy.
It's not anywhere under their wastewater treatment thing. And I was like, I know it's there because I've been there. Like, it's near one of my sites because, that's why I think it's polluting there. I literally drive past it. Their gates are always closed. Trucks come out, but I can never come in. I called the city building across from them and said, hey, can I please reach out to this building? We don't have contact for them. Oh my gosh. I was like, why are y'all impossible to talk to? It's like, do you want this thing to be bad? No, are you scared?
They're like, ignorance is bliss. Let's just ignore it.
If we don't know it exists, we don't have to spend any money on fixing it, and stuff can just die, and Lake Hamilton can keep being green and ugly.
Yeah, if it's doing like the gravity stuff, like that's kind of a big deal. That's kind of a big thing, like that helps the city function. So you would think there would be like people over that. Who's the contact? There's got to be someone.
Yeah, but I couldn't find anything. I was really looking at like the address. I kept searching like Hot Springs Lift Station. It won't show up on Google Maps. Like I went on Street View and I looked at the sign to make sure I wasn't crazy, because I was like, I know this exists.
Yeah. So if you're like listening, like, and you know any contacts, if you're like, if your dad or mom
works for Hot Springs, let me know.
Please give Kira Burnett contact. In the info on this episode, like I'll have some info how to contact.
Yeah, you can put my number. This is urgent.
Wow. I was like, well, I don't know if I can put her number, but
you can put my number.
It is serious, guys. Guys, we need to save the water streams and like the creeks and the river, the Watchtower River.
If you have somebody that works in like the Environmental Protection Agency, let me know.
Please. Oh my gosh. So if you want to go to ASMSA and do some like serious work and not know who to tell, go to ASMSA. I don't know. How should I close this up?
Do you want to talk about like research in a park? Because I've already talked a lot about my project, and how serious the issue is.
Yeah, it's like really sad, actually. It's like...
Are we going to talk about science fair?
Okay, science fair. Yes, we're going to talk about science fair.
So we have a regional science fair because we're composed of so many schools, like so many sending schools. So we have the...
No, I actually didn't know what I was going to say. I'm sorry.
You're good. So we have the West Central Regional Science Fair. And so everyone in ASMSA competes, everyone in a STEM capstone competes, either their junior or senior year. If you're in research in the park, you do both. So I competed both years. And first, second, and third place, and sometimes honorable mentions, will go to the State Science Fair, and from State Science Fair, then you can go to ISEF. We also send 3 people, the overall 3 winners, to ISEF as well. And you get money for if you win first, second, or third, or if you win those first, second, third overalls. And then we also have special awards from different organizations, such as like Regeneron, the people that run ISEF, the Stockholm Junior Water Prize. I won that.
Oh!
NASA, we have a NASA award.
Air Force.
Yeah, Air Force, Yale, Missouri S&T is a big one. I won that one last year. Also Magic Springs, but they didn't show up this year. I was really gunning for that one. I thought that'd be funny.
Because you're like water.
Well, I was just thinking it would be funny to have one from Magic Springs. It was just for like an upstanding project for the Hot Springs community, and I was like, that's so funny. Like, who won there's one for the U.S. metric system. If you use the metric system in your project, you can do it. I think it was Riley Ramick that won it, and I was like, bro, I wanted that one so bad.
Yes, Riley.
It just says metric system.
Like, I know you want that. Like, that's cool.
Seriously, I was like, that's a funny award. I want that one.
Like, I remember being at the award ceremony and we just keep clapping. There's like award after award after award. I mean,
yes.
It goes on for like ever. It's so many awards. It's like amazing.
So there's a lot of chances for you to win.
Yes. Go to science fair. I say as I'm not going to science fair. I'm humanities. But I would love to. It was really cool. So many cool projects. People are like dressed up fancy and they're yapping. They're, like, good project.
We have the first day for like participants, we go in there, set it up on Wednesday of the science fair week, and then on Thursday we will have like the day of interviews. So you'll have 3 judges usually. You can have more, like animal sciences or chemistry are our largest categories usually, and so they have like 4 to 5 judges. It's really just whoever can come. It can be ASMSA parents. My mom did it. It can be alumni. It can be anyone they can reach out to. And then later that day, you will hear back if you get the ISEF interview, which means you have been selected as one of the top like projects, and you have to come back and get a second interview to a new team of judges. It composed of 2 this year. I got an ISEF interview.
Whoop, whoop.
So there were 6 projects that got it. So I was top 6 projects overall. I unfortunately did not proceed. I was not one of the top 3 here.
Very competitive.
Yes, it's very competitive. I was really surprised. I was actually eating with my mom, and then I got the email. I was like, Mom, go back right now. I ran up there. I was in like bummy clothing. Munson told me to go back and change. But it's a very stressful event overall. But then that next day, the Friday, is when you have public viewing. So other students can visit, parents, like anybody can come in off the street and come see your project. A lot of people come in, like different, like even judges from other categories will come in and say hi and like see about your project if they were interested the day before.
Nice.
In the morning, and then in the evening is the award ceremony, which is like what everyone's waiting for. That's when you get to know if you won or if you lost or if you're going to state.
Yeah, what you got like first in your category, right?
Yes, I got first in my category this year, which meant I won $100 in a Walmart gift card. I also get to go to state. And then I won the Stockholm Junior Water Project Prize, which no money, but you get to go to a state competition. It's online, but if you win that state competition, then you go to nationals, which is like anywhere in the country, and you get like a full paid expense trip.
See, that would be fun.
I really hope I get it, but I mean, I don't know who else has won it. It's all online, so it's like I don't get to really see anything. I don't get to go there. Like state is in person, so like there I can walk around, I can assess other projects and like
see your
my
competition.
Chances. Yeah, see my competition and be like, oh, am I gonna win it this year or not?
Be like, well, theirs kind of sucks, so I think I'll be good.
That's how it was. That's how I thought it was last year for state. And then this kid in my category, I won't say his name. I'll keep him anonymized. But he got on 100 on his score sheet, and it was the first 100 they'd ever given out in 10
Oh my
years.
gosh.
And I was like,
That's impressive.
I was like, that's awesome, but also I'm cooking. That's the first thing they announced in the state ceremonies. I was like, oh man, that kid's in my category.
So it's like, I'm done. Like,
yeah,
he already beat me.
I knew it right then. I ended up getting second last year, but I was like, oh dang it.
But for a junior, I feel like that's really good.
Oh yeah, we're so far we've been the best junior class of Research in Park.
Ooh.
Because we all went to state. That's the first time I think it's ever happened for Dodd. Waddell's been here a little bit longer. She's the other Research in the Park advisor. Yes, we're the best class. We all went to state. And then the three finalists for this year at regional that went straight to ISEF, two of them were Research in the Park students in my class.
Yes. Colton and Lucas, right?
Yes. Colton and Lucas, their project, and then Christine Dunning as well.
Yes. So, I mean,
Shout out to my crew. If you're in Research in the Park, you'll win.
Yes. No, but seriously, I mean, Dr. Dodd and Waddell push all. And speaking of that, I had a note, boot camp, or what do y'all do? What do y'all do the weekend before? Nora's told me about it.
Okay, so it's a ceremonious activity called RIP Boot Camp, where the weekend before science fair, you'll go in at 8 a.m. I've been there at 7 a.m.
Why would you do that?
I needed to work, man. I needed to get to work. But they'll bring in food, donuts, cheese.
I love cheese.
Fruit. Dr. Dodd brought in her own sourdough bread. It gets very fancy. And then you will work for an upwards of 10 to 12 hours that day.
Each day? Oh my.
No, just on Saturday. Sundays, if you're a bum and you have to come back. It happens though. I came back on Sunday.
Oh.
But usually it's 10 to 12 hours, and you can come back on Sunday if you didn't get it done. But it's just getting everything finalized. Poster number 1 is the thing that get finalized. Mm-hmm. Quad chart, which is just a quick summary that they have you print out at your poster. They have slideshows this year. That was a new thing.
Slideshows?
Yes, you had to do a slideshow because ISEF finalists, they have to have, once they become an ISEF finalist, they have to have that ready and completed a week after our science fair. So they just made everyone go ahead and do it. And also for judges to, preview your project, because usually we'll submit our papers into, the electronic science fair website called Z-Fairs. Usually we'll do our papers, but this year they said, no, put your slideshow and put your quad chart. So it's a shortened version. Hold on, I forgot what I was talking about. Oh, RIP Bootcamp, yes.
Yeah.
Sorry.
You're good.
So for RIP Bootcamp, we work on completing all these components and getting them all done. It's painful. It's tiring. It's just you'll have everything complete, you think, and then you wait yeah, and then you wait 30 minutes, and then you get a review back, and it's 40, 50 comments.
Oh.
And you have to fix everything. And then towards the end, it's so infuriating because it'll be you just need to fix 3 things, but everyone is waiting to get those reviewed by Dr. Dodd or Waddell, and so
you have to wait the line.
Yep. I this year came into boot camp even though most of my stuff was done. I got my poster printed in 3 hours, I think. I came in because I'm also the biology lab peer mentor, which is I just do whatever Dodd needs to assist, and I've been helping with students' projects throughout the year, helping revise papers. And so I was the one looking over the posters, trying to clean that line up faster, because a lot of people just have grammatical errors or making it sound better. Things like that, and so I helped accelerate that process. I think I helped a lot.
I mean, nice. RIP got a lot of awards at science fair. Yes. Dr. Dodd was so happy. She was handing those awards out, and she was so happy. I was happy for her.
Yes, we we also have a taxidermy groundhog named Morty.
Yeah.
He also comes to our science fairs, and he's in the classroom, and he's a big help for Research in the Park. He's sort of our mascot. And so there's definitely photos of me holding Morty at my poster like a baby.
That's beautiful. I love that. So yeah, if you want to join RIP, community, there's going to be RIP boot camp. And you have to do park work service, the National Park Work Service.
Yes.
which is lit. I do it, and I'm not in RIP. I just like it.
Yes, your junior year, you have to complete National Park Work Service. If you're unfamiliar with ASMSA, all students have to do like 30 to 40 hours of work service.
30 hours per semester.
30 hours of semester.
Which now across Arkansas, from like the LEARNS Act or something, like
that's normal now.
Yeah, so it's kind of,
you already had to do that, but now like ASMSA, that's been a long-standing tradition.
yeah.
And so if you're in research in the park your junior year, you have to complete National Park Work Service, which, if you don't know, Hot Springs is a national park. We're in a national park. And so you go help the national park by like ripping out invasive plants. You can do water quality with them of the springs, like water quality sampling. We do a huge trash cleanup every year.
And didn't we like win this year? Or we got a trophy.
I didn't get to show up. I was sick this year. But last year we got second place. But then they still gave us this trophy because they're like, come on, we're kids. Like the other people you're competing against are like old retired people.
That's kind of funny. I will say though, recently, as of May 4th, which is, I don't know when I'm publishing this, but as of May 4th, 2025, the National Park
May
visitor
4th or March 4th?
March, sorry. Okay. So tomorrow. March 4th, the visitor center will be closed Tuesday and Wednesday because our national park now only has 3 staff members.
Hey, Trump
Park
did
rangers.
That. I read this article. I'm sorry. I won't say that. It's true though.
Kira Burnett. Oh. Well, you know, stuff happened. We now have less park rangers, and I'm a bit worried about our work service. That's a personal thing.
Yeah, I have zero clue what's happening. That somebody asked me earlier because I run the greenhouse work service as of the moment, and people were asking me like, "Well, I'd love to help you, but like, do you know if National Park Work Service is coming back?" And I was like, or like, is it going to continue? I have zero clue. It's a little harrowing and scary to see.
It's very like, I don't know, because our work service is normally on Wednesdays, and now they're being closed Tuesday and Wednesday.
So I don't know if it'll be moved. If it's Friday, a lot of people will be gone. If it's
Monday, it's not always a B day.
Exactly.
Because B days is when we get out early at 2:30. We wouldn't have time if it was 4:15 because they normally close at 4.
Yeah, exactly.
So basically, you'd never know what your work service will be. If not, it'll probably be garden or
greenhouse.
Yeah.
We have a new greenhouse now. We're having the first native plant cell this year. I'm running that now with Adame. He's part of the environmental science class, and in that class you have to do some sort of service project, and so he's helping me with that now. But we have a greenhouse, so a lot of opportunities there. We're just growing plants that we've taken from the garden or things we've bought like seed wise, but you could grow food, you could grow like fruits and vegetables possibly.
Because you're doing like sunflowers right now, or?
Yeah, we have a mix of a ton of plants that are in our garden. We have a ASMSA native garden ran by Don Waddell. And so we have foxglove beard tongue, which is this ornamental white flower bush. Bees really like it. We have Maximilian sunflower, which is like huge. It gets like 8 feet. Huge sunflowers. We have Plains coreopsis, which are like small yellow flowers with like a brown red center. We have a variety. I'm trying to think of other things. Anise hyssop. I don't know really exactly what it is. I just got the seeds. I was excited to see it come up soon. And we just got violets, and we had another endemic plant come in today. Ozark something. Ozark phlox maybe. Ozark prairie phlox. So we have a lot of variety in the greenhouse, and if you come here, it's open to a lot of things, because I don't know if this plant garden is going to be annual, because if I'm not here, who's going to run it? It might be something passed down to the next generation. I don't know. This is the first year. Or it could be a place. Yeah, the next generation of ASMSA. Or it could be for like fruit and vegetables or anything you want to grow. You could use it for your capstone project. Students started doing that this year.
And then, like, y'all have like a garden website, right?
Yes, I do not remember the link off the top of my head.
I will find it and I will put it in the description.
Yes, but you can find plenty of information about the plants in our garden, sightings, who's worked on it. We have multiple features in the garden that students have built over the years. Last year, me and a recent graduate, Ella Campbell, we built a Shishiadoshi fountain, which is also called a deer scare garden. You might have seen it if you went to Disneyland. They're at Disneyland. But it's like it's a bamboo fountain that will like fill up the bamboo shoot with water, and then it will like clack down, and it's supposed to generate noise to scare deer off from your garden. We also have a deck built by students last semester. We have a new wall put in, huge rocks, and a step like stairwell, or like a set of stairs and a walkway. So a lot of opportunities to build onto this garden. All that information is on our website.
Yeah, I forgot how big RIP is. Like RIP has so many different like elements, and like it's definitely like a full experience. Like
yes, and it's even if you're not as committed to research in the park, or let's say you come in as a student and you really want to do something else, and then you're like, I miss out on this opportunity. There's environmental science, which is a class where you're required to do a service project. There's also garden work service, of course, as mentioned, greenhouse work service. So you could be participating in these activities any way you want, but research in the park is truly like the whole package.
You know, I was talking to like one of the RIP kids yesterday, and they said something about like an outdoor classroom development.
Oh, yes. Okay, so we started the garden through research in the park. So during COVID, it was harder to get our students off campus. They were only allowed to be off campus for a couple hours a week, and then so the work service thing got harder to arrange, and so they decided they really wanted the space on campus where they could interact with native plants and, wildlife. So we've got a lot of insects attracted to the native plants, and so pollinators, basically. So we started that garden, and that was in 2020 that we started applying. We got a grant through the Smithsonian, actually, by competing in something called the Teen Earth Optimism Competition through Mid-America, which is an affiliate of the Smithsonian. And so, yeah, that was just $500, but it got us going, and we held on to that and applied for more grants. And there are so many grants out there for starting native plant gardens, so we've just kept expanding.
Yeah, it's huge. Like every single, like, I would say quarter, like I see something new pop up.
Yeah.
And, like,
absolutely. And the outdoor classroom is part of that. So we had a Zen garden, these are all kind of student ideas that was proposed a couple years ago. And so we got that in place after Selig was renovated, and that space kind of opened up, and we saw that it was pretty depressing and there wasn't much there because they really just worked on the inside of the building. Yeah. So the students said we want to do a Zen garden with a sand bed and a rock garden and moss. There's this student that had done a project on moss and wanted to add those plants to the garden, and so we did, and we had shade beds that we're trying out and things like that. But yeah, the outdoor classroom, the idea came to build this. We wanted to build like a structure for shade and rain protection so people could be out there in all weather. But so far we've just got the platform, but this spring we're going to add some benches and / tables, and then maybe in a future year we'll add the roof.
Oh, okay, cool. Yeah.
So yeah, that's what that is. But yeah, the garden, I mean, we've got so many different parts. Like in the past year or so, we expanded with the students created a Diana fritillary garden, the state butterfly, with plants that would attract that butterfly, a monarch garden. We added a checkerboard garden this fall. So there's some new features out there. So yeah, whatever students are interested in doing. That's kind of within the research in the park class in the fall, and then environmental science in the spring.
Cool. So yeah, thank you for coming on the show.
Thank you so much for having me. I was so excited to come on.
Yeah, and see y'all next week.